Hello and welcome to BioCode. In this video, we will see how we can utilize Entrez module from BioPython package to do a summary research on any record, any sort of data set, any sort of database within NCBI. So in this video, what we are going to accomplish is we are going to utilize utilize an ID of a research paper, and that is on of a review paper on application of sport vector machines in cancer genomics, which is a great paper by the way, and you should read it if you have some time. So I'm going to utilize its PMID, PubMed ID, and we are going to extract information out of it through BioPython. And we just need to utilize the eSummary function. eSummary function basically retrieves a summary of a particular ID that you provide, of a particular query that you provide. For example, if you have a gene name, if you have a gene ID, if you have an accession ID, you can put that in by specifying a database to search it against. For example, if you're inputting gene, then you should always select that database to be gene, otherwise it will not work. So we are providing an ID of a review paper. Then we are searching against PubMed database, and we are asking for the summary, and we are not actually retrieving the entire paper. We are not retrieving this paper. We are retrieving information about it, summary about it, and then we are reading that handle. So this will be fairly same throughout the series, so I'm not going to discuss about this. Then we are printing the entire record, the record that we actually see, that the information that is retrieved through NCPI. So I'm just going to run this code, and we will see some various information that you, there we go. So this is all the information about the paper, and you may have seen some hanged or some slow internet connection there. This is not a slow internet connection, some sort of that kind of thing, but it really depends upon your internet connection, but not so much. First, this request has to be sent to the NCBI database, then the information is retrieved, so it will take some time before the result is actually shown to you. Anyways, so a list of dictionaries is retrieved, and it will contain a dictionary such as item, ID. This is the same ID, as you can see, same ID, and a PubMed date, EPUB date, source of the paper where this research paper or review paper is present. Author list is also present, which is a list of authors, and title is also present. Applications of sport vectors, SVM, learning and genomics. Let's see if it is same. There we go, it is same. And then we have the volume, issue number, pages, and that sort of information. We do have a DOI as well, and a plethora of information is available depending upon the ID and depending upon the journal. Anyways, what we are going to do, we are going to extract title, journal name, which is source, and then we are extracting DOI as well, digital object identifier. So I'm just going to do a string formatting. We are saying journal ID info should be, we are saying journal info should be, first we will retrieve the ID. We don't need the ID, we just need the source. There we go. First we need the source or the name of the journal, cancer genomics, proteomics. Then the full name of the journal is also available, which is cancer genomics and proteomics under full journal name, which is basically a key. Since it is a list of dictionaries, so that dictionary should always have keys and values. So each of these keys can be used to retrieve a particular set of value or information. So I'm just going to extract source, then we are going to extract title, and then we are going to extract DOI as well. So same through this. Since this is the first element, so we are using the 0 because it only has a single, this list should always have a single list of dictionaries, right? There is just one dictionary within this list, but this dictionary contains various other elements in it, and we are just utilizing, for example, we are just utilizing the keys to extract that information. We are utilizing source, title, and DOI. So let's rerun it, and let's see if the information actually comes out. There we go. We now have source, cancer genomics proteomics, title is also available, application of support vector machine learning in genomics, and DUI is also available. So that was fairly easy to retrieve this information. You can play around and extract more information as well. I'm just going to do it. What I'm going to do is I'm going to input some other paper's ID. Then we are going to retrieve information about that. So this is the ID. Let's switch it over here. Let's run our code and let's see what happens now. There we go. Title changed, source also changed, DOI also changed. So this is fairly easy to extract information about any of the paper that you want. You can do the same set of information. You can extract different information utilizing same set of code. So I'm going to extract information about a particular protein, and this time it will be information about the summary of the protein, not the entire protein sequence or that sort of information. We are just going to extract summary about this protein. So we are utilizing the database to be protein. We are passing passing the ID or the accession ID of the protein through a variable protein accession. Then we are reading it and then we are printing out the entire code. I'm just going to make this comment so this is not printed again. Okay, so let's run our code, and this time it will print out the summary about the protein that we are searching.